Hi everyone, welcome back to PW International Boards on this series of one shot that will be very useful for your upcoming examination. In previous session, we have recognized the characteristics of all living organisms. We also named the organism scientifically using binomial system. We were able to classify the organism based on some characteristic features. All right. In this session, we will talk about the structure of plant cell, the structure of animal cell, the structure of bacterial cell, and how these structure perform their function. We will be talking about the magnification. From that, we have observed at least one question in each paper that has come from magnification topic. So, according to that, this today's session is very important to you all. All right, let's begin with our session here. So, our first slide is about the topics to be covered in particular session here. That is the cell structure, in which we will be taking the plant cell, all right, the animal cell and bacterial cell. All right, we will study about their structures as well. Then size of specimens will be taken as another important topic that include many questions from different question papers. We have already added not few, so around 50 or more than 50 questions from past papers considering these two topics. So you can consider that this particular session is very important. All right. So all organisms are made of cells. Every organism has a living cell in it. All right. So cell is the basic, functional, structural and fundamental unit of life. We have the defined here as the cell is the basic, fundamental, structural and functional unit of life. So we have this diagrammatic representation. Let's not get very much confused about what are these structures. We are going to study these structures in this session only. The organisms can be defined on the basis of whether they have single cell as the whole body or they have many cells in their body. So we have unicellular organisms, we have multicellular organisms. Unicellular organisms are those organisms that have single cell in their body and they function as a whole organism. Talking about the multicellular organism, there are many cells and all cells perform different functions to keep that body alive. All right. Then we have the examples for these. Bacteria is the unicellular organism. Then we have yeast as the unicellular organism. Examples of multicellular organism, we have plant cell, we have an animal cell as well. All right. So to observe the cell structure, we will require types of sections. These are two types of sections, longitudinal section and the transverse section. When we cut along the length of the stem, this will result in the longitudinal section of that particular stem. And when we cut across the structure, this will result in a transverse section of the stem. So we have just considered the part of stem here. We can take any tissue, an animal tissue, plant tissue, leaf, stem, branch, root, any of the organ can be taken out to have this particular section. All right. So whenever we are going to cut the plant structure, so there is a normal technique that we can use, a normal razor blade for that. So razor blade will be useful in cutting the plant structure. So thin sections of the plant structures can be obtained using a razor blade. You can observe leaves, you can observe roots and stems as well. All right. So you can make the thin slices of each organ that you need to study. Then we have, if you need to consider the cutting sections of animals, so first what will happen, the animal tissues are soft and flexible. So we need to soak them in melted wax for quite some time, and when they become solid, you can easily cut out thin sections of the animal tissues and we can study further. So the wax will dissolve away whenever we are studying about the particular thin slice of that animal tissue. All right. So we have the animal tissues that need to be soaked in melted wax before cutting thin sections, as they are very soft and flexible, and once they get solidified we can cut them easily. Whenever we talk about the microscopic examination of plant cells, so cells can be seen We can see the cells using the microscopes, all right. So we have a comparative diagrammatic representation here. On the left side there is representation of animal cell, and on the right side we have the plant cell. So both of these have some common cell structures. These are nucleus. So nucleus will be present on the animal cell as well as in the plant cell. We have the cell membrane, that is the first boundary to any cell. So animal cell has the cell membrane, plant cell also has the cell membrane. Then we have cytoplasm, which is present in both. So in addition to these cell structures, plant cells do have another cell structures, which are the cell wall, which are the permanent vacuole and the chloroplast. So we are going to study about all these structures in this session. Animal cells, they are much smaller than the plant cells. So all right, animal cells are much smaller, so we need to magnify them more to see them, all right. So stains are often used to enhance visibility of internal structures within these cells. So we use stains or we use specific dyes to enhance the visibility of internal structures within these cells whenever we talk about the animal cells, all right. So the human eye cannot see most of the cells, all right. The hand lens magnifies about 10 times. The cells can be often seen as dots. Yet we are unable to see the cell internal structures by this kind of magnification here, all right. The light microscope magnifies 400 times to 1500 times. That is a huge times of magnification. With a light microscope you can see some structures inside a cell, such as a nucleus, or you can see the other structures as well. An electron microscope magnifies 40,000 times to 5 lakh times. With an electron microscope much more detail can be seen. So you can see that how the observation can be carried out using the different kind of microscopes here, all right. The basic components that are present in almost all living cells are cell membrane, nucleus and cytoplasm. So let us understand what are these structures first, since these are the most common components that are present in almost all living cells. And why we write almost word here? Because the nucleus will be absent in the bacterial cell, and we are going to study about that also, all right. So considering a cell, let us draw a cell. So while drawing a cell, we will first make a boundary. All right. So this first boundary here is the cell membrane. All right. This is the cell membrane. Then we are going to make a round structure over here. This here represents nucleus. This is nucleus. All right. So here nucleus is there. And what about the remaining structure here? This region. So this is the cytoplasm, all right. So this is the cytoplasmic region in a cell, all right. This is the cytoplasm in a cell. We have just started to become an artist. All right. So here we have the cytoplasm. All right. So these are the structures which will be present in almost all type of living cells. We have the diagrammatic representation. So there is this first boundary. This is the cell membrane. All right. So all of these cells here represented having the cell membrane. Then we have this nucleus, all right. So this is the nuclear region. Then the shaded part here is the cytoplasm, all right. So this is the cytoplasm here, all right. Now moving further, we are going to discuss about them in detail, all right. So here we have All right. So components of cells are there. We have the cell membrane, plasma membrane. So we can call the cell membrane as the plasma membrane as well. So it is the thin layer surrounding the cell. All right. So this is about the plasma membrane. So it is a very thin layer that surrounds the cell. All right. The plasma membrane, or you can also call this as cell membrane, it is a lipid bilayer basically. We have these hydrophobic tails. So these tails are of fatty acids. All right. Then we have the hydrophilic head, and these head are of phosphate. So these are phosphate heads. Phosphate head here. And by this you can observe that this particular diagram here represents all of these structures. So these, the hydrophilic head of phosphate, all right, and to the inner of these we have the hydrophobic tails, and these tails are of fatty acids. So this is particularly a phospholipid. This is a phospholipid here, all right, and we have the lipid bilayer here. Lipid bilayer, in which the fatty acid tails project inward. These are the hydrophobic part of lipid bilayer. So these are hydrophobic in nature. Hydrophobic. All right. Then we have these phosphate heads. And these are hydrophilic. They face the aqueous environment. All right. So these phosphate heads are surrounded with aqueous environment, and that is the reason why we have kept the hydrophilic part on the outer side, and toward the inner side we have kept the hydrophobic part. All right. So functions of the plasma membrane are very important, and we can observe many questions have come through the direct statements as well. So here we have our first statement that says it acts as a barrier. So it prevents the cell contents from escaping. So it let them bound together in the cytoplasm and provides a boundary to the cell. So by this it will prevent the cell contents from escaping. All right. Then we have, there is selective permeability, which is shown by the plasma membrane. So it is a property by which only certain substances are allowed to cross the cell membrane, or you can say plasma membrane. All right. Then we have controls which substances can enter and the substances that can leave the cell. It is always controlled by the cell membrane here. Then we have intake of oxygen will be there, nutrients will also be required. So oxygen will be required by the cell to carry out the chemical reactions that require oxygen as the substrate. All right. So nutrients are also required. Then we have water. So cell is mainly made up of 70% of water. All right. So water is required by the cells. Then we have hormones and etc. So all of these components here are provided an entry into the cell, and what is removed from the cell, so removal of waste products is there from the cell, and it includes carbon dioxide as well. All right. So carbon dioxide will be produced by the chemical reactions as the product, and it will be, since it is a toxic product, so it will be removed out from the cell. And how does this exit and entry of oxygen and carbon dioxide takes place? So this is controlled by the plasma membrane here. All right. Then it maintains the overall shape and integrity of the cell. So basically when we are about to draw a cell, so we first draw a boundary. So the boundary defines the shape of the cell. All right. And this boundary here is the cell membrane. We are talking about the cell membrane, which is able to provide this cell its overall shape most of the times. All right. And when we consider the plant cell, there is another structure that is responsible for the same and provides the rigidity to the cell also. In animal cell, we can observe that this semi permeable membrane here, or you can say selectively permeable membrane, or you can say partially permeable membrane, is the cell membrane which is responsible for maintaining the overall shape and integrity of the cell. It facilitates chemical reactions within the cytoplasm since it allows entry and exit of certain substances across the cell membrane. All right, moving to the next slide here, we have, all right, so the first structure was the cell membrane or the plasma membrane, and then we have come to the second structure, that is nucleus. So here we have the nucleus representation. All right, so let me take to the previous slide here. This one structure here is the nucleus. All right, so this nucleus has been magnified, and there is also a cross section showing the internal part of the nucleus as well. These are the nuclear pores here. Then we have the nuclear membrane. So nucleus has double membrane. All right, we have the chromatin threads here. The chromatin threads are condensed to form the chromosomes, which are only be seen during the cell division of a cell. All right, we are going to study about that also. Then we have nuclear plasma, there is nucleolus also. So what is the structure saying here? Most cells contain one nucleus. So why does it say that most cells contain one nucleus? Why did not say that all of the cells contain one nucleus? So there is this feature of we can say polyploidy, in which cells have more than one nucleus. All right, so that's why we have taken this statement that most cells contain one nucleus only. The nucleus appear as a rounded structure enclosed in a membrane. So nucleus is appearing as a rounded structure enclosed in a membrane. All right. This structure is also fixed in the cytoplasm. Fixed in the cytoplasm. All right. It may appear darker than the cytoplasm due to differential staining, because the nucleus takes up certain stains, takes up certain stains more strongly than the cytoplasm, and that is the reason you can say that the nucleus are densely stained regions in a cell. So I am writing it here. Nucleus are densely stained regions in a cell. It may appear darker than the cytoplasm due to differential staining, and I have already made you understood how this happened. Then functions of the nucleus are, this controls the enzymatic activity. The nucleus regulates the type and quantity of enzymes produced by the cytoplasm. So it is main, we can say main structure here, which is responsible for controlling the enzymatic action in the cytoplasm. So it regulates the type, so with which type of the reaction will be, and the quantity of enzymes that will be produced is controlled by the nucleus. So you can say that nucleus is having very much heavy duty here, and it is basically acting as the brain of the cell. All right, then it act in the chemical regulation. It oversees the chemical changes occurring within the cells. So it keeps a check on the overall chemical reactions, chemical changes occurring in a cell. I'm so sorry. So all of these functions are being performed by nucleus. Then we have the cell identity feature. The nucleus determines the cell's identity, that is whether the cell will be a blood cell, whether the cell will be a liver cell, muscle cell, nerve cell. So the identity is decided by the nucleus. So you can write it here in a simplified language that this cell's identity will be decided by the nucleus. All right. The nucleus has another function, that is the cell division. The nucleus controls the cell division. All right. So nucleus controls the cell division. It is the most important point over here. And you can also say the nucleus controls the cell division. Yes, we are going to study about the cell division also. I have added the slide over here in the upcoming slides as well. All right. So we will study about that also. Then we have the chromosomes, which are thread-like structures containing genetic material DNA and RNA, are present in the nucleus. So DNA are the structures, these are basically thread-like structures present in the nuclear region that are going to be visible during the cell division process only. So let me write it here that chromosomes are visible during cell division only. And here you have this diagram in which a cell is dividing. The nuclear content is also dividing, and nucleus is able to control the cell division as well. All right. The cytoplasm is the another cell structure after nucleus, after the cell membrane, which will be common to all of the living cells. All right. Then we have the features of cell cytoplasm here. It contains thick liquid with visible particles. All right. So it is a thick liquid, or you can say it looks like a thick liquid, looks like a thick liquid with particles in it. All right. So with visible particles, or you can say particles embedded in it. All right. So you can observe these particles whenever you use the light microscope. So the cytoplasm is visible. You can use a light microscope to see cytoplasm and particles in it too. All right, in it. Then we have another feature that the liquid component is approximately 90% of water. So we have just took this data that 70% or 90% of the cell volume is the liquid component, and this liquid component is the part of cytoplasm. And what are the solid component or the particles which are present in the cytoplasm? So we have the list of those two. These are organelles. We are going to study about the cell organelles as well. Dissolved molecules. Dissolved molecules are also present in the cytoplasm as the solid components. Then we have salts, sugars. Food reserves are also there in the form of oil droplets, starch granules, larger molecules of fats, lipids. So fats, either you can say fats are there, either you can say lipids are there. So this should be a slash over here. All right. So fats or lipids and proteins etc. are present as the particles in the liquid component of the cytoplasm. So this is about the cytoplasm. We have this diagrammatic representation in which cell membrane is there. So here we have this region as the cell membrane. All right. This is, all right. Let me just correct it. Here we have nucleus, and in nucleus we have nucleolus. There is this cytosol, and what is cytosol? The liquid component of the cytoplasm surrounding the intracellular organelles. And by intracellular we mean that all the cellular organelles which are present in the cell. So by intra we mean within the cell, and by Inter, we mean in between the cell. So two cells and the region between it is termed as the intercellular. And when we talk about the intracellular region, so it is the region that is present in the cell only. All right, so talking about the intracellular organelles, so these are the organelles which are present in the cells. All right, I hope this terminology is clear to you all. Then you have this cytoplasm. All right, so contents of a cell except the nucleus includes all organelles as well as the cytosol. So we have just discussed about that. Then the functions of cytoplasm are huge in number. We have, it serves as the region for chemical reactions. So large number of chemical reactions occur in the cytoplasm. So there is something synthesized by the cell organelle, which is then transported to the other cell organelle, which then consumes it and convert it into another structure, which is then consumed by the other part of the cytoplasm here, or you can say other cell organism. So there is this pathway occurring in the cell where one region is synthesizing, other is modifier, the other region is the consumer here. So all of these reactions are occurring in the cytoplasm, because cytoplasm is the region that serves as the medium between one cell organelle and connecting it with the another cell organelle. All right, so large number of chemical reactions occur in the cytoplasm. All right, then we have the reactions provide energy. Now how do these cell performs this process of synthesizing energy? So energy is required by the cell to perform the process of cell division, in which the cell divides. So by the division of the cells, it requires a lot of energy, and all of the reactions that are responsible for providing energy occur in the cytoplasm as well. We will study about the structure which is responsible for synthesizing energy in a cell. All right, then we have synthesize necessary substances for the cells, and then we have just discussed that one cell structure is able to produce something, the other cell structure is able to modify it, or you can say that it directly consumes that structure because it is necessary for its function. All right, then we have, it provides the region for enzymatic activities as well. Enzymes control the rate and type of chemical reactions in cells. So what are enzymes? Enzymes are, let me write it here. Enzymes are, I had already discussed about enzymes in the previous slide, or we can say previous chapter discussion as well. So enzyme are proteinaceous in nature. These act as biocatalyst. That means they increases the rate of chemical reaction. They remain unchanged throughout a chemical reaction. Throughout a chemical reaction. All right. So they remain as such throughout a chemical reaction. Some enzymes are attached to cell membranes, others float freely in the cytoplasm. So there are enzymes which are present as an attachment to the cell membrane, and others are there that float freely in the cytoplasm as well. Now another structure we have is the cell wall. So cell wall is not present in all living cells. They are specifically present in plant cells, in fungi, in bacterial cells. All right. So we have this diagrammatic representation of cell wall structure. So the cell wall mainly composed of the cellulose, and if we consider different cells, so the composition of cell wall in different cells will be different as well. In plants it is mainly composed of cellulose, and along with that we also have another components as well. Hemicellulose is there, pectin is there, all right, and proteins are also there to form this cell wall structure. The middle lamella is there, primary cell wall, plasma or cell membrane is also represented here. All right. Cellulose microfibril is there. So we will just have this overview that how a cell wall structure looks like, alright. Beginning with the features and the functions of cell wall. So it contains cellulose and other compounds. So what are these other compounds? We have just found out that hemicellulose is there, pectin is there, protein is there. So we will write the other compounds as well. Here we have hemicellulose, pectin, proteins as well, alright. It is a non-living structure. It is not something like the cell membrane. It is a non-living structure. The cell membrane was living structure. This one is a non-living structure. It is not selective like the cell membrane also. So the permeability of the cell membrane is that it shows selective permeability, or you can say it is partially permeable in nature. But the cell wall is not selective like the cell membrane. The plant cells have a both of these, a cell wall and a cell membrane, and we have this structure to make you visualize where the cell wall and the cell membrane is present in a plant cell. But before that, we can also draw this diagram here, that so this is a plant cell over here. I have just made one boundary, and inner to it we have another boundary. This is the cell membrane which I am making through the white marker here, and the green marker here represents the cell wall. It has nucleus, all right. It has majority of its region occupied by vacuole, so 90%, up to 90%, all right, not 90%, up to 90%. So each cell has different type of volumes that are occupied by the vacuole present in them. So this is a general diagrammatic representation of a normal typical plant cell, all right. I have not considered the chloroplast here because the chloroplast will be present in those plant cells that are able to photosynthesize, and by photosynthesis they are able to make their own food material using raw materials from outside, all right. Let me label it for you. This region here represents cell wall. We have this region as cell membrane. You will also observe many questions coming from this particular diagram, that you have to label parts of a plant cell. Where is the cell wall? Where is the cell membrane? Where is the vacuole present? Where is the cytoplasm? You have to identify the nucleus, chloroplast. So we are going to solve those questions in this class itself, all right. Then this region here represents nucleus, and this here represents vacuole, all right. So the region here is the cytoplasm. I hope this is clear to you all. So plant cells are distinct and easy to see due to their cell walls. You are able to see the plant cells easily. Plant cells are generally larger in size when compared it with the animal cells. So you are able to see the plant cells easily due to the presence of their cell walls, all right. And on the other hand, the animal cells being smaller in size, they do not have the cell wall, so it will be quite not easy for these animal cell to be seen. We need the magnification to be higher in that case, all right. Then we have another function, that it allows water. It is not living structure. The cell wall is a non-living structure, but it allows water and dissolved substances to pass through it, all right. We have come to this section of cell organelles. So we are consider this particular cell organelle that is present in both animal cell and plant cell. But what difference does it serve in both of these cells? So in animal cell, the most of the region is not occupied by vacuole, whereas in the plant cell it occupies 90% of the cell volume, or you can say a range of 70 to 90% is occupied by the vacuole in a plant cell's volume. And when we talk about the animal cells, so they may have the vacuole, they may not have the vacuole, or they synthesize the vacuoles whenever they require, all right. So we can say that vacuoles are prominently seen in plant cells. So we have this representation here Other cell organelles are also there. Alright, we have the mitochondria. This is the powerhouse of the cells. It generates energy for the cell to carry out the process of cell division or other metabolic reactions as well. Then we have cell wall. We have just understood that this is the outer membrane or outer covering or outer layer that is present at the outermost layer. All right, then we have inner to this is the cell membrane, and it is not represented here, so let me just mention it here that this is the cell membrane. All right, we have the nucleus as well here. Then there is plastids. So plastids are the cell organelles that are present in, again we will talk about that comparing the plant cells as well as the animal cells. All right, then we have this vacuole. It has a boundary, it has this slight boundary over here. All right, so this is the membrane of vacuole that is termed as tonoplast. So we will write the functions of the cell organelles here itself. Mitochondria is there, that is the powerhouse of the cell. Then you have got here the cell membrane. It is selectively permeable in nature. Alright, then you have got here the cell wall. This is the non-living structure of a plant cell. The nucleus, let me write that it is the brain of the cell. It controls the reactions occurring in the cell. We have the plastids. So there are different type of plastids that are responsible for providing or imparting different type of colors to the plant cells. All right, then we have vacuole occupies 80 to 90% of cell volume in plants. And we have just understood about the cytoplasm. So it is the main region or arena for the cellular activities. Now, it is a large fluid filled space. So it is a large fluid filled space here, found in most mature plants. All right, so why it is not present in all plant cells, we will also study about that also. All right, so it contains the cell sap, that is, so what is the cell sap that is present in the vacuole? So it is a solution of sugars, salts and pigments that constitute the cell sap, which is then present in the vacuole. All right, so here we have the cell sap located here. So this region here is having the cell sap. Animal cells may have small vacuoles in their cytoplasm, or they might not have the vacuole, so they will synthesize them whenever they require it. Pushes the, so it is the function of vacuole that it pushes the cytoplasm outwards. So by the presence of cell sap in the vacuole, so it will exert some pressure. All right, it will exert the pressure and it will push the cytoplasm outwards. It is surrounding the cytoplasm here. So imagine if this is a vacuole here and surrounding this there is the cytoplasm. By the presence of cell sap here, this will be able to push the cytoplasm outward and creating a thin lining inside the cell wall. So that is also understood here. Maintains the cell turgor pressure, and by the presence of cell sap in the vacuole, there will be turgor pressure that will be generated in the cell due to the presence of vacuole. All right, so contributing to the firmness of plant cells and tissues. So it is able to contribute or maintain the firmness of plant cells and tissues as well. I hope you understood this one also. Then we have the cell organelles in which, all right, so there is representation of vacuoles here. All right, so here we have these structures are vacuoles. All of these are vacuoles. And we have, alright, there are also vacuole. These all are vacuoles here. You can observe that on the left hand side, the cells are having more kind of vacuole, or by that we can say that there is more amount of cell sap in the vacuoles on the left hand side cells, and these cells are turgid, all right, due to the presence of cell sap. On the right hand side, we have seen that these cells are having reduced vacuole, and also the cell contents are shrinking away from the cell wall here, all right. So the cytoplasm has become reduced in size, and it has concentrated toward the inner side of the cell here. It has moved away from the cell wall. So by this we can say that this is a representation of plasmolysed cell. We will study about the plasmolysis, we will study about the cell being turgid, we will study about the cell being flaccid in the another section, or we can say another session of this one shot, all right. The another cell organelle here is the mitochondria. So mitochondria is the powerhouse of the cells. It generates the maximum amount of energy that is required by a cell. So whenever we are saying that the cell is requiring much amount of ATP or much amount of energy, that means that it has a huge amount of mitochondria in it. So mitochondria are responsible for energy production. Or you can also say that this energy is in the form of ATP. And what do we mean by ATP? So what is the full form of ATP? Here we have adenosine triphosphate, and it is formed by joining inorganic phosphate to ADP. ADP I said. So D is for adenosine diphosphate. So by adding the inorganic phosphate to adenosine triphosphate, this requires a lot of energy, and this energy is then stored in the bonds of ATP. So from ADP, adenosine diphosphate, we have made the ATP, adenosine triphosphate, all right. By that we have stored the energy in the chemical bonds of this ATP molecule, all right. So all right, so mitochondria are circular, oval, slipper shaped organelles, all right, when we see them in the cut sections, or you can see the lateral views as well. So in three dimension, they may be spherical, they may be rod like, or extended. So whenever we considering the 3D structure of mitochondria, we are talking about that the structure looks like somewhat spherical, somewhat rod like, or extended. So it is rod like and extended, so it is basically a sausage shaped structure. They have a double membrane structure, all right. So here we have the outer membrane, this is the outer membrane here, and inner to this is the inner membrane. That has enfoldings. So these are the enfoldings, all right, so as to increase the surface area for ATP generation. The more enfoldings there, the more will be surface area for the reaction of evolving the energy from the mitochondria, and more will be the energy production by these enfoldings. And they have the double membrane structure, we have talked about this. Then its outer membrane is permeable to all substances. So let me write it here that it is permeable. Outer membrane is permeable. The inner membrane with many inward pointing folds so as to increase the surface area. So as to increase the surface area for ATP generation. Over here we have another statement that mitochondria are most frequent in region of rapid chemical activity. So these will be present in those regions, and though also present in abundant quantity where there is rapid chemical activity occurring inside the cells. All right, then we have they release energy from food substances. I will make you understand what this statement means actually. Through the process of aerobic respiration. We have already understood about the respiration in the previous chapter as well. So let us get a recall about that. So respiration is a process in which you get the stored energy released from the bonds of the food molecules, and this process occurs through two ways. Either you can utilize oxygen for that process to release energy, all right, or you can carry out this process in the absence of energy as well. So by utilizing the oxygen to break down the chemical bonds of carbohydrate molecule, this process occurs in the mitochondria, and since it requires oxygen for the process, that means that it is an aerobic type of respiration. All right, so breakdown of food breakdown of food molecules in order to release energy, in order to release stored energy. All right, so in order to release this stored energy, there is this process of respiration. It occurs through two ways. It can either be aerobic respiration, aerobic respiration, or anaerobic respiration. All right, so by the aerobic respiration we mean that to carry out this respiration process it requires oxygen. This anaerobic respiration do not require oxygen to perform this respiration. And how does this happen? Considering mitochondria as the intermediate region for the process of aerobic respiration. So what actually happens here? Here we have the glucose molecule. So it is C6H12O6. So by a 10-step reaction, that is the glycolysis process, it is broken down into pyruvate. All right. This pyruvate is then entering into the mitochondria. Mitochondria then acts on the pyruvate molecule, performs the process of aerobic respiration, that is, it utilizes the oxygen to release ATP. All right. So this is what we mean by the aerobic respiration, which is performed by the mitochondria. All right. So conversion of glucose to pyruvate do not require oxygen. So this is kind of a process that occurs in all type of cells, whether they do aerobic respiration or they do anaerobic respiration. So this step is common in all type of cells, and the glucose to pyruvate conversion happens in the cytoplasm region, by which the mitochondria receives the pyruvate and performs the aerobic respiration to carry out the further process. All right, I hope you understood this one also. So here we have the cell organelles. We have studied about the cell structure, that is the cell membrane. It is selectively permeable. So by these slides we are also memorizing each of the term we have just studied in this session only. All right. Then we have the cytoplasm where all the cellular activities will occur. Then we have ribosomes on membrane. So ribosomes are those structures that are responsible for protein synthesis. So they are also known as protein factory. All right. Then we have nucleus, nuclear pore, and the mitochondrial region. So let me give you a brief about the ribosome as well. So here we have ribosome. Synthesizes protein, hence called as the protein factory. So present in both eukaryotic cell and prokaryotic cell. All right. So it is also present in let me take this space here. So also present in cell structures which are, now you have to keep this in mind. All right. So this is chloroplast. All right. Then it is also present in mitochondria. So these are the two cell structures which have the ribosomes in them. All right. Along with the ribosomes, the chloroplast and mitochondria also have. So let me write the star mark point over this region. Chloroplast, mitochondria have their own DNA, ribosomes. That means they are semi-autonomous in nature, semi-autonomous organelles. Organelles, all right. So this is a special point about these two cell structures considering chloroplast and mitochondria cell structures. All right. So they have their own DNA, they have their own ribosomes. All right. And what are ribosomes? Ribosomes are the structures that are responsible for the synthesis of protein. All right. These are both present in eukaryotic and prokaryotic cells as well, or we can say that they are also present in the cell structures which are chloroplast and mitochondria. Moving to the next slide here. So we have come to another cell organelle, that is the chloroplast. Now what do we mean by chloroplast? These are the type of cell organelles. Alright, so let me first make you understand that these are found only in the photosynthesizing parts. Alright, so present in regions that perform the process of the process of photosynthesis. All right, and by photosynthesis, what do we mean? We mean that fixation of, or you can say the substrates, carbon dioxide and water in the presence of sunlight by the chlorophyll pigment converted into carbohydrate plus you can say that oxygen molecules are also evolved. So this is the reaction for photosynthesis process. All right, so carbon dioxide and water are required for this process, acted upon by the sunlight, converted into the carbohydrate and oxygen molecules by the chlorophyll pigment, and these chlorophyll pigments are green in color. So these are green pigments. So chloroplasts have green pigments in them, and these pigments are of chlorophyll. A chloroplast is also a double membranous structure. So we have double membrane structure. Since we have just understood that it also has its own, so this is about has its own DNA and ribosomes. So here we have the diagrammatic representation of chloroplast. So this is the outer membrane here that has been cut to make you visualize what is present inside of this chloroplast structure. So this is the outer membrane here. We have inner to this is the inner membrane. All right. Then we have the structure. So this is a thylakoid. This is a thylakoid. This whole structure refers to a granum. All right. It has its own DNA. It has ribosomes. You can also observe these are the ribosomes here. These are the ribosomes. All right. Then chloroplasts are the organelles found in plant cells. So these are found in plant cells, and those structures specifically which are able to photosynthesize. All right. Then we have, they contain the green pigment called as the chlorophyll. We have just understood about this one also. Chlorophyll absorbs the energy from sunlight, convert it into chemical energy. All right. So let me complete this sentence over here, that chlorophyll absorbs energy from sunlight and converts it into chemical energy. All right. So we have the light energy, and the source for light energy is, you can guess this right. So this is sun, the ultimate source of energy. All right. You can also observe many questions coming directly from this statement, that what is the ultimate source of energy. So that we have sun for that. The carbon dioxide is required, the water is required, and these chloroplasts are present in the leaf structure. Since the leaf are the green structures, or you can also say that those structures in a plant that appear to be green in color are able to photosynthesize. Whether it is the young stem, it will also photosynthesize if it is green in color. So the green pigment is basically provided by the chlorophyll pigment present in chloroplast region. So these carbon dioxide and water are required by the chlorophyll. On the action of sunlight, it is then converted into the glucose here, so that we have C6H12O6 over here, and oxygen molecule is also released. So glucose here is stored in the form of starch in the leaves as well. All right. This energy is used for photosynthesis. So chlorophyll absorbs the sunlight energy. All right. So this energy, and by this we mean that the sunlight energy. The sunlight energy is used for the process of photosynthesis. We have understood about the photosynthesis and where does this occur, how the process happens or takes place, where plants produce food, and food in the form of a carbohydrate. And to be specific, we have understood about the production of glucose here. So chloroplasts are absent in animals since they do not photosynthesize. They cannot make their own food from the raw materials here. Then we have most plant cells in green plants like leaves have chloroplast. So it has stated that most plant cells. So why does it stated that the most plant cells have the chloroplast? Since there are certain plant cells that do not photosynthesize. They carry out their specific function but do not perform photosynthesis. For example, you can consider a root hair cell that is specialized for performing the function of absorption of water and minerals from the soil. All right, it is not forming the function of photosynthesis, so it will not be present in the chloroplast. So chloroplast will not be present in the root hair cell. All right, then we have chloroplast often contains starch grains produced during the photosynthesis. So starch grain are produced since these are the stored form of carbohydrate that is the product of photosynthesis. All right. We have the different type of cells. This is prokaryotic cell, and then we have eukaryotic cell. All right. So we have certain pointers to that, and prior to these pointers, let me make you understand that the prokaryotic cells These are the primitive type of cells. They lack any membrane bound organelles. All right. So they have ribosomes. Ribosomes. They, you can say that they lack any membrane bound organelles. They have ribosomes. They have all right. They have poorly developed nuclear region. So you can say that nucleus is not present, but that a poorly developed region is present, that is termed as, if you all can see. So, all right. This is a nucleoid. Alright, so they have poorly developed nuclear region called as a nucleoid. They do not have nucleus. They lack any nucleus. Any nucleus. Alright. Okay. Then we have, you can say that the, alright. So let us compare the prokaryotic cell and eukaryotic cell over here. So the example for the prokaryotic cell is a bacterium, and for the eukaryotic cell we have the plant cells. Alright. So the prokaryotic cell do have cell wall as present in the plant cell, but the composition is quite different. All right. So, all right, you can say that here there is this genetic material just scattered in the cytoplasm. Genetic material scattered in cytoplasm. And you can observe that there is no membrane of nucleus here. So nucleus is not there, yet you can say that there is a poorly developed nuclear region that is termed as nucleoid. And one question also have come from this particular statement, that whether the prokaryotic cells or the bacterium cells have the nucleus or not, or you are provided with different kind of cells in which you have to select out the common cell structures which are present in all the type of cells, and you will get the nucleus as an option. So you will mark nucleus as an as a structure that is not present in prokaryote, you can say bacteria. All right. So here you can also observe that there is the presence of flagellum. So flagella is not present in plant cell here. All right. Flagella is a structure that enables the cell to move. All right. And in comparison, we have the plant cell, that is the eukaryotic cell. So most of the cell organelles are present in eukaryotic cells, but the prokaryotic cell do not have any membrane bound cell organelles. They do have ribosome. Ribosome do not have any membranes. All right. So prokaryotic cells, or you can say bacterial cell, now since we have considered that bacterial cell is an example of prokaryotic cell. Bacteria are very small unicellular organism. Their length is less than 0.01 millimeter. That is, if you convert the unit here, so this comes out to be 0.01 into 10 to power minus, what just happened, so this is -3 meter in length. All right, they can only be visible under highly magnified microscope. So to visualize the bacteria, you require a highly magnified microscope. The structure is there. We have the capsule, that is the coat of the bacteria here. Then you have the cell wall. So cell wall is there. The plasma membrane is there. Plasmid. So plasmid are the structures all right. So plasmid DNA are the structures that enables the bacteria to be used in the genetic modification process, since it is easy to ligate a foreign gene or a segment of DNA to this plasmid DNA, and it multiplies and gives us the required gene required protein as the product. All right. So cytoplasm is there, flagellum is there. So it is present in some of the bacteria. Not all of the bacteria have flagellum. So those who require movement require the presence of flagellum. Then we have nucleotide, that is the DNA. So this is a genetic material. They do have ribosomes. All right. So pili are also there. So it is basically having a function in the DNA transfer as well. Then we have the cell wall is made of a mixture of proteins, sugars and fats. Since we have already discussed that the cell wall will be present in plant cells, fungal cells, bacterial cells, but the composition will be different in all of these cells. Plant cells has the cell wall that has cellulose as the component, whereas talking about the bacterial cell, it has proteins, sugars and fats as the composition. Cytoplasm contains a large number of ribosomes. So ribosomes are the protein factory. Glycogen granules are also there. Fat and other food reserves are also present. Ribosomes synthesize the proteins and hence you can call them that. Ribosomes are protein factory of the cell. Alright, so there is again a one diagrammatic representation in which it is clearly stated that the flagellum is present in some bacterium only. Alright. Glycogen granule is there. We have the ribosomes. Cell wall is also there. Alright. Then we have chromosome single DNA strand coiled up. Cytoplasm is also there. Inner to the cell wall, we can observe that it has the cell membrane. Now comparing the eukaryotic cells. So for the eukaryotic cell, they are the true cells that have nucleus, that have membrane bound cell organelles as the structures present in the cytoplasm. And for that we have the two examples. Animal cell is there, plant cell is there. Both of these cells are examples of eukaryotic cell. The difference between the plant cell and animal cells, here we have the ribosome, here we have the cell membrane. Alright, so it is not clearly mentioned that this is the cell membrane. Alright, let me mention here. Nucleus is there, cytoplasm is there, mitochondrion is there. Then we have chloroplast, which is not present in the animal cell. Then it has a cell wall of cellulose, which is also present in plant cell only, not found in animal cell. Vacuole is there, so vacuole occupies 70 to 80% of the cell volume in plant cell, and animal cell may or may not have the vacuole. Cytoplasm, ribosome, so alright. Now we have the table here that represents the difference among plant cell and animal cell, and we have observed many questions coming from this table directly, but you could not remember them directly. We had to discuss about the cell structures, we had to discuss about the functions of the cell structures to memorize and to be able to differentiate whether this is a plant cell, whether it is an animal cell. So to visualize a plant cell, you can simply identify the presence of cell wall and you can call that this is a plant cell basically. Alright. So for that we have the plant cells. First feature for that is have a cellulosic cell wall outside the cell membrane. So there is a cell membrane, then you have the cell wall outer to this. Alright. The animal cells do not have cell wall. We will mark tick for the quite of satisfaction that we have just gained after discussing about the cell structures, the cell types, and you will be able to recall all of the features in this particular table. Alright. So have a cell membrane, both of these have cell membrane. Alright. Cytoplasm is there in both. Have a nucleus, both of these have nucleus. The plant cell often have chloroplast. So why did we say that they often have? Why did not we say that all of the plant cell have the chloroplast? So all of the cell in a plant cell do not perform the process of photosynthesis. So that's why we have just stated that often have chloroplast containing chlorophyll. As stated, animal cells do not have chloroplast. Often have large vacuoles containing cell sap, and by cell sap they are able to make the cell turgid. They we can say push the cell push cytoplasm to the outward. So vacuole is there, then it is surrounded by the cytoplasm, so it is able to push the cytoplasm outward due to the presence of cell sap in it. have only small vacuoles, or we can say might be absent in animal cells, often have starch granules, never have starch grains, sometimes have glycogen granules. So the stored form of carbohydrate in animals is starch, that is stored in leaves. The stored form of carbohydrate in animals is glycogen, and it is stored in the liver, all right. Then we have often regular in shape due to the presence of cell wall that provides a rigidity to the cell. Often irregular in shape when we talk about the animal cell, because they do not have any cell wall. So, all right. Endoplasmic reticulum is there. So, it is a structure that is present in close association with the nucleus. When there are ribosomes attached to the endoplasmic reticulum, we say that it is responsible for production of protein. And whenever there are no ribosome attached to the surface of endoplasmic reticulum, then we say that this particular reticulum is a smooth endoplasmic reticulum, which is further responsible for synthesis of lipids. So let me just write it here for a quick revision. We have smooth endoplasmic reticulum, do not have attached ribosomes responsible for lipid synthesis. Then talking about the, all right, so you can use this space over here, rough endoplasmic reticulum. So due to the presence of ribosomes on its surface, it is able to produce proteins. So have ribosomes on it. And synthesizes proteins. All right. I hope it is visible. All right. Synthesizes proteins. Nucleolus is there, nucleus, mitochondria, peroxisome, chloroplast, ribosome, Golgi apparatus, cell membrane, cell wall, chloroplast, vacuole, amyloplast, cytoplasm. So we have just discussed about the cell structures that are given in our textbook, all right. So moving further, we have the mitochondrion, lysosome, cytoplasm, centriole. So centriole are the structures that are visible during the you can say, so these are responsible for the cell division process, all right. Cell membrane is there, ribosome, smooth endoplasmic reticulum, nucleus, nucleolus, rough endoplasmic reticulum, Golgi complex. So it is the apparatus that is basically performing the modification and packaging of the proteins and lipids synthesized by the smooth and rough endoplasmic reticulum. The cell division is a process by which a single cell gives rise to two daughter cells, or you can here see that the bacterial cell is present in the A diagram. The cytoplasm divides since the nucleus has already divided into these two. There will be no nucleus in the bacterial cell, so we will simply say that the genetic material has been divided into two daughter cells here. The cytoplasm is still attached here. The cytoplasm then divides, then we say the cell has divided. The each cell here will divides through the process, you can say the cell division then again occurs. Then you have this section of specialization of cells. So specialization of cells occurs, all right, all right. So we will write about it here. The specialization of cells occur The specialization of cell occurs to make the cell more specialized for a particular function. They attain a specific structure, they become mature after the cell has divided. Now the cell has a particular structure, a particular function to be performed, and for that we have different type of cells that are present in this particular chapter. We are going to study about the ciliated cell, root hair cells, xylem vessels, palisade mesophyll cells, nerve cells, red blood cells, sperm cells and egg cells in this upcoming slides also. So we will first discuss about the structures, where it is found and what are their functions, and then we will proceed with this table. So it will be more easy for us to recollect our information and get the table understood in a easy way. So we have the ciliated cell over here. Alright. So ciliated cells are the cells that forms lining of nose and windpipe. Alright, so these are the cells that form the lining of nose and windpipe. So these are the cilia here. These are the cilia. These hair-like extensions. Alright, these are the cilia. Then talking about the feature of ciliated cells, these are tiny cytoplasmic hair called cilia. Alright. Then you have another feature that the cilia shows a continuous flicking movement, shows a continuous flicking movement. And what do we mean by the continuous flicking movement? So they are able to create a stream of fluid. All right, we will write about it as well. By flicking movement, all right, they are able to create a stream of fluid, that is basically mucus when we talk about the trachea and windpipe. So it enables this mucus to be moved out from the lungs through the bronchi and the trachea. So we have create a stream of fluid, that is mucus, that carries dust and bacteria through bronchi and trachea away from lungs, all right. So these are the features when we talk about the ciliated cells. These form the lining of nose and windpipe. All right, the tiny structures, the cytoplasmic hair, are known as the cilia by the flicking movement. So let me just make you understand how this flicking movement occurs. So let us consider that these structures are a cell. All right. So these are the two cells. They have their extensions in the form of cilia. So what they will do, they will perform a coordinated movement. All right. So they will all be in a coordinated movement. So this movement is a continuous flicking movement. By this, they will generate a stream of fluid that is to be pushed along the direction of this flicking movement. All right. So considering the mucus to be removed out from the lungs through the bronchi and trachea. So let us imagine that there is this lungs over here, the cilias are there, so they will perform this flicking movement to push the mucus out. All right, so this is the kind of coordinated movement. All right, I hope this is understood to you all. All right, then we have the root hair cell. So root hair cell is responsible for the absorption of water and minerals from the soil. All right, they extend into the soil particles. They have this extended structure here. These extensions are root hair that help the plant to penetrate, or you can say they help the root to penetrate deep into the soil to grab more and more water and mineral ions for the absorption process and to be carried away toward these cells from the roots. And we can write the features over here. So the root hair have very thin wall. All right, so let us talk about that. The root hair cells are responsible for absorption of water and mineral ions from soil. All right, we will again talk about the root hair. So the hair penetrate between soil particles, and you can say, and offers a large surface area for absorption. All right, the cell membrane of root hair cell. So root, I will write it here like cell membrane of root hair cell controls entry of certain dissolved substances into the cell. Substances into the cell. All right, so I hope you understood this pointers as well. So these are very important as per examination point of view. All right, so let us understand the structure over here. We have the structure here, the hair penetrate between the soil particles. So let us imagine that these structures, the root hair, are surrounded by the soil particles. All right, so they penetrate deep into the soil. The particles are pink colored. All right, the soil, I'm representing here the particles by this pink color. So these are soil particles. Did you ever see the pink soil? You just see it here. We have identified the pink soil. Apart from this, all right, so these are the soil particles. All right, soil particles around it. So absorption of water and mineral ions will occur through these root hair cells, all right, from the soil. they extend between the soil particles and offers a large surface area for absorption. So why is this hair like structure is present in root hair or root cell? So basically if they extend the structure, they will extend the surface area. The more the surface area, the more will be absorption of mineral ions and the water from the soil. So this is the function of root hair cell. Then we have the cell membrane structure here. So cell membrane of root hair cell control entry of certain dissolved substances into the cell. Alright, we have the root hair cell as the specialized cell. Then we have here the palisade mesophyll cells. Alright. So they have the cell wall. Alright. So since it is a plant cell, alright, let me write it here. Since it is a plant cell, since it is a plant cell, cell wall will be there, will be present. Alright. Now, so where can you observe these palisade mesophyll cells? So let us imagine that this is a leaf. Why to imagine? I have already tried to make the leaf structure over here. So on magnification of this section of leaf, you will observe that this structure contains palisade cells in them. So parallelly arranged cells which are responsible for carrying out the process of photosynthesis. These cells have chloroplast in them. Alright. Numerous chloroplasts to carry out the process of photosynthesis. They have nucleus. They have nucleus in them. They have vacuole as well. So vacuole is also there. All right. Magnified view. All right. I hope it is clear to you all. Then coming back to the diagrammatic representation here, we have the nuclear membrane. All right. So this is the nuclear membrane over here. We have the nucleus. There is representation of nucleus over here. Cytoplasm is there, vacuole is there, and chloroplast. So chloroplasts are the cell organelles that have chlorophyll as a pigment in them. Have chlorophyll in them. So by the presence of chlorophyll, it will be able to photosynthesize. It is a specialized cell. Since it has become specialized to perform a special function, that is to carry out the process of photosynthesis. Alright. So we can write the pointers for palisade mesophyll cells. I will utilize the space present over this region. Alright. So these cells are found underneath of upper epidermis of plant leaf. All right. Talking about this structure, they are columnar cells packed with chloroplasts. So palisade cells are columnar cells packed with chloroplast. With Chloroplast. Now, due to the presence of chloroplast, it is able to perform the function of photosynthesis. Due to this, it performs photosynthesis. All right, you can observe it. Done. So this is all about the palisade mesophyll cell. Then we have the specialized cell, that is an egg cell. So that is the female sex cell. All right. We will write about it here as well. Or you can say female gamete called as ovum. Alright, so these structure here represents an ovum, that is also called as an egg cell. Alright, for that they also have this feature of you can say that they have the right. So we have the cytoplasm containing yolk droplets. All right. So cytoplasm containing yolk droplets, these are mainly composed of fats and proteins. So let me write it here that the yolk droplets are made of fats and lipids. All right. These ovum are spherical in structure. Spherical in shape. All right. So let us go through the diagram. We have here the jelly coat of the egg cell, which is digested by the male sperm cell. So that is the male sex cell that has a region of acrosome in the tip of the head that has digestive enzymes to digest the jelly coat of the egg cell here. All right, and by that it is further you can say it is further proceeding towards the fusion of the male gamete with the female gamete, leading to process of fertilization. All right, so let us take an example over here of the all right, so let me draw an sperm cell here. I will utilize this space. We have the head region, then you have the middle piece. It further extends to form a tail or flagellum that enables it to swim. We have in the head a region of acrosome. We have in the head nucleus. We have in the mid piece, that is mitochondria are there to provide energy for swimming, and we have the tail over here. So this is the tail. All right. In this region, this is representing acrosome. Acrosome is there. All right. Then this region represents nucleus. We have the mitochondria here. So these are mitochondria. We have the tail present in this region. So this is the tail. All right. Let me mark the regions as well. So This region is of head. Alright, so I will mention it here that this is head region. This one is the mid piece. So there is this diagrammatic representation of a sperm cell in which you can identify the acrosome is there, nucleus is there. So acrosome has digestive enzyme. I will utilize the space over this region. Has digestive enzyme to digest jelly coat of an egg cell. Alright, so it is responsible for this feature. Then we have mitochondria. These are responsible for providing energy for swimming, provides energy for swimming. The tail is behaving as flagellum. Flagellum for whip-like movement. Whip-like movement. Alright, so this is about the head region, the mid piece and the tail. Alright. Coming back to the ovum here, it is the female sex cell, then it is called as ovum. It is spherical in shape. It has yolk droplets that are made of fats and lipids. So this is another specialized cell. Moving toward the RBC, that has a very distinct feature, that is that makes this cell very different from the other cells, and that is the absence of, so mark my words, absence of nucleus. Yes, nucleus is not present in mature RBC. So don't be surprised whenever this term pops up in the question as well. So whenever the question says that mark or identify the cell that do not have nucleus in its mature phase, so you are going to answer that RBC is that one cell. So absence of nucleus in mature RBC. All right. So RBCs are disc shaped cells. So I will write it here. These are tiny disc shaped cells. The RBCs contain pigments called hemoglobin. So hemoglobin is there as a pigment. All right. Now this pigment enables the RBC to perform the function of transportation of oxygen throughout the body. Since these hemoglobin bind to oxygen molecules and carry them along the whole human body. So we have hemoglobin that performs the function of transportation of oxygen by binding to them. So binds to oxygen and performs the function of their transportation. Across the body, across the whole body. So this is about the RBCs. So cytoplasm contain hemoglobin. The cytoplasm has hemoglobin. Hemoglobin binds to oxygen, and by that it is able to transport the oxygen throughout the human body. All right. I hope this is understood to you all. Then we have another cell here, that is the nerve cell. All right. So nerve cells are able to conduct electrical impulses, along the fiber to and from the brain and spinal cord. So it involves the CNS. CNS is the central nervous system, and in central nervous system it has brain and spinal cord. So conduct electrical impulses along the fibers to and from the brain and spinal cord. So to and from the brain and spinal cord. So talking about the fibers are often very long, and it connects different parts of the body to CNS. And what does CNS include here? CNS has spinal cord, plus brain. All right. Then coming back to the diagram here, we have this cell body. So all of the body here except the nucleus that has the cytoplasm, all right, it has nacelle granules as well. So this structure here is the cell body. All right, this all structure here excluding the nucleus part. All right, this is the cell body. Okay, then we have the nucleus. In this region nucleus is present. We have the nerve fiber that serves the function of conducting the nerve impulses, or you can say electrical impulses, and this carries the electrical impulses from the brain to the brain, from the spinal cord and to the spinal cord as well. There is also one other another point to this, that chemical reactions cause the impulses to travel along the fiber. We have chemical impulses causes the chemical reactions. Chemical reactions causes the impulses. Reactions causes the impulses to travel along the fibers. All right. So these structures here represents dendrites. We have the endings here. These are the nerve endings having, or you can say that nerve endings this particular strand of cytoplasm, or you can say the cell body that extends throughout this nerve fiber, is termed as the axon. All right, now moving to the another topic, that is the magnification. So there have been multiple questions coming from this topic, and it is very important as per examination. So a lens is marked with magnifying power, indicating how much larger the image will appear compared to the specimen's actual size. So we are basically comparing the larger image to the specimen actual size. So we are comparing the magnified image with the actual size of the particular object, or you can say organism or cell. All right, then we have, for example, if a lens is marked at 10 times, all right, then the image will be 10 times larger than the real size of the specimen. So this has marked the magnification on it, that it is showing the image of the we can say object or the cell or the specimen that has been magnified to 10 times, or you can see the magnified value that is being taken from the microscope. All right, so how to calculate the magnification? We have the defined here to calculate the total magnification. So first of all, what do we mean by magnification? So we have to tell about that first. Magnification, it tells a comparison between the magnified value and the original value. Or you can also say that magnification do tell us about the how many times the larger image is when it is compared to the actual size of the cell. All right, we have the first defined here. To calculate the total magnification, multiply the magnification of both lenses. So if the magnification is present, so you can write that the magnification can be calculated by using this formula, that you have eyepiece magnification multiplied by the objective magnification. That is, for instance, if you use a 10 time eyepiece lens and a 40 time objective lens, that you have to multiply both of these magnifications to get the total magnification. All right, so you will get 400 times of total magnification by this. When you draw an image, it's usually larger than the actual image seen through the microscope. That's why we use the microscope now. So microscope are the structures. Microscope are basically used to observe those structures that are not visible to the naked eyes. All right, so therefore the overall magnification of the specimen is even greater than the calculated value when we hand draw any cell or any image. So when you draw an image, it's usually larger than the actual image seen through the microscope. So whenever you are drawing any image, you are basically already magnifying it to many times, since that particular structure was not able to be visualized by our naked eyes. We use a microscope to visualize that particular structure. For that, we also have the formula to calculate the magnification, and you will solve the questions based on magnification concept and the formula in today's class as well. So magnification is equals to the observed size of the image or the drawing. If drawing is there, so we will write the size of the drawing on the numerator side, and then in the actual size of the specimen will be written in the denominator size. Or you can also say, on rearranging the formula, that the actual size of the specimen is equal to the observed size of the image divided by magnification. And this particular section is very important, and you will observe one question coming directly from the formula, that you have to mention the correct formula for magnification from the given options. You have to tick the right option basically. This is very important. Now, the things you need to keep in mind while calculating the magnification is that ensure that the units for both the actual size and the observed size are the same. So, keeping the units both, basically if the question is saying that the image has we can say the size is there in millimeter, so we will keep the size in millimeter for the actual size and for the magnified size as well. All right. If they differ, actual size in millimeter and observed size in centimeter. So if there is a different units taken for the measurements here, then convert one to match the other. So you have to convert the other to match the other. So basically if you need to calculate the answer, then you have to convert the actual size in centimeter either, or you can do the observed size conversion into the millimeter. You can either convert the actual size from millimeter to centimeter, or you can convert the observed size from centimeter to millimeter also, keeping both the units same for the observed size and the actual size. Remember that there are 10 millimeters in a centimeter. So they have already stated that if you ever encounter any question that has centimeter as a unit, so you will write it that 1 centimeter has 10 millimeter. All right. Alright, so we have come to the questions part of the topics we have discussed as per examination view. So get ready to solve this along with me, and it will be really, you can see that the questions are directly getting solved by the concepts we have just discussed. Alright, so we have the first question here that says, the image shows an animal. So we have this diagrammatic representation of an animal. The length of the animal in the image is 36 millimeter. So this is the length of the image. Image length here is 36 millimeter. Then you have actual size. All right, so actual size is also provided. 12 millimeter. You have to calculate the magnification of the image. We have just discussed about the magnification formula, and for that we use the formula as magnification equals to magnified value by original value. Keeping both the units of actual and image sizes in same. So magnified value divided by original value. What do we have as the magnified value here? It is 36 millimeter. We have the actual size as 12 millimeter, so we will directly get the magnification as 3 times. B option is right. So these questions are taken from past year papers. So you can see how easy the questions has become after understanding the concept thoroughly through these sessions only. All right. Now we have the second question that says the diagram shows a plant cell. So we have this diagrammatic representation. Since we have already discussed the plant cells, we can relate that this here represents root hair cell. Root hair cell, which features show that it is a plant cell. So plant cell have cell wall that is not present in animal cell. The plant cell have a vacuole. So vacuole amount is different in different type of cells. So in plant it occupies up to 90% of the cell volume, whereas in animals it might be present or may not be present or be synthesized whenever these vacuoles are required by the cell. So which features show that it is a plant cell? It has a cell wall, it has a vacuole, and first option suits well. So A is the right answer. It has a nucleus and cytoplasm. It is present in each type of cells, though nucleus will be absent in bacterial cells. So this does not state that the given cell here represents a plant cell only. All right. Then we have, it has a nucleus but no chloroplast. That is true, but this do not state that it is different from other cells. All right. It has chloroplast but no vacuole. This is incorrect. Question number 3 says the diagrams show 4 different type of cells. Which cells contain hemoglobin? And we have just discussed that hemoglobin is present in red blood cells. We have to identify which of the given options resembles the red blood cell. So A here is the, this is the root hair cell. B is a palisade mesophyll cell. D is a sperm cell, and C here represents RBC. So RBC have hemoglobin. We have question number 4 that says which adaptive feature of sperm provides the energy that enables it to swim? So the energy is provided by the mitochondria. Alright. So since it is a powerhouse of the cell, mitochondria is able to generate ATP. That is the energy. Alright. And by this, it is enabling the sperm cell to swim. The diagram shows a fly. We have this diagrammatic representation. The line XY represent length of the wing. So this diagram is a magnified size of the actual size of a fly. The length of line XY is 26 millimeter. So this here is 26 millimeter. The actual size of the wing is 4 millimeter. Alright. So you can write it here. Magnified value is 26 millimeter. Original value as 4 millimeter. You can get the magnification by calculating these two. Magnification is, what you're gonna do, you're gonna put the values over here. So we have magnified value as 26 millimeter, keeping the units the same for both of these values, divided by 4 millimeter. You will get an answer that is 6.5 times. All right. So B option is the right one. Next we have question number 6 that says, all right. So keeping the enthusiasm high as we have to solve a lot of questions in this session. So question number 6 says which structure is present in plant cell but absent in bacterial cell? So you can say that bacterial cell do not have nucleus but plant cell do have. Alright. So cell membrane will be present in both, cell wall will be present in both, cytoplasm will be present in both, but nucleus will be present in plant cell only. Question number 7 says which cell is adapted for swimming? And for swimming we have just discussed about the sperm cell. So C here represent a sperm cell. This is the right option here. Though A here represent palisade mesophyll cell, then we have B that represents ciliated cell. Alright, we have D that represents root hair cell. Question number 8 says a pollen grain was viewed under a microscope using 500 magnification. So we are already provided with magnification here. Magnification is 500 times. The image of the pollen grain was measured as a 75 millimeter. And whenever we talk about the image size, we are going to relate it with the magnified value. So we are provided with magnified value as well. So we are going to find the actual size that comes out to be on dividing these two. So we have the actual size of the image equals to magnified value divided by the magnification. So magnified value here is 75 and the magnification is 500. Now going back to the options, we get option A as the right answer. Question number 9 here says, which diagram represents a cell responsible for transport of oxygen? So we have discussed that for the transport of oxygen, hemoglobin is required. So hemoglobin is a pigment that binds to the oxygen molecules and transport it across the body. It is present in RBC. Present in RBC. And A here represents the RBC, alright. Moving to question number 10 that says the list shows the sum of the structures contained in cells. So we have the cell structures here: cytoplasm, cell membrane, cell wall, chloroplast. How many of these structures are found in cells of all living organisms? We are going to select those structures that are present in all living organisms. Cytoplasm is there? Yes. Cell membrane is there? Yes. Cell wall is not present in animal cell. Chloroplast will be present in only those plant cells that photosynthesize. So we have 1 and 2. Option B is the right answer. Question number 11 says a student draws a red blood cell. The diameter of red blood cell they draw is 20 millimeter. So we have got the magnified value as 20 millimeter. The actual diameter of the red blood cell is, alright, actual value 0.008 millimeter. Since the units are same, we do not need to convert it into any other unit. What is the magnification here? It equals to magnified value by actual value, that comes out to be 20 divided by 0.008 millimeter. Now solving this we get, alright, so 20 into 10 to power 3 divided by 8 over here. So this is 2.5 into 1000. This is 2500. So magnification comes out to be 2500 times. Option D is the right answer. Question number 12 says a diagram shows a plant cell in which labeled part of the cell is glucose made. So you have to recollect your information about the process of photosynthesis. So by photosynthesis process, glucose is made by chlorophyll in chloroplast. So you have to identify the region that represents chloroplast structure. So A here is vacuole, B here is chloroplast, D is nucleus, cell wall. Alright, moving to next question. So question number 13 says the diagram shows an epidermal cell from an onion cell. The distance between X and Y on the diagram is 60 millimeter. So we have got the magnified value. Magnified value here is 60 millimeter. You are provided with the actual size of the cell. Alright, so that is 150 micrometer. Now you need to convert the units. Alright, so let us just focus how do we solve this question. Magnification is magnified value divided by actual value, or you can say original value. Magnified value is 60 millimeter. Actual value is 150 micrometer. Alright. So 60 into 10 to power minus 3 meters, since 1 millimeter equals to 10 to power minus 3 meter, then you have here 150 into 10 to power minus 6 meter. So let me write it here. The conversions we used. we used here that 1 millimeter equals to 10 to power minus 3 meter. We use that 1 micrometer equals to 10 to power minus 6 meter here. All right. Coming back to solving this, we get 60 by 150 into 10 to power 3. All right. So this comes out to be 6,000 divided by 15. This is 400. Yes, this is 400 times. Magnification comes out to be 400 times, and C answer is the accurate one here. All right. We have question number 14 that says some components of cells are listed. Cell membrane, cell wall, chloroplast, cytoplasm. Which cell components are present in plant cells but absent in animal cells? So cell membrane will be present in both of these cells. Cell wall is present in plant cell only. Chloroplast will be present in plant cell only if we are considering that plant cell which is photosynthesizing. Cytoplasm will be present in both of these cells. So which components are present in plant cell but absent in animal cell? We have second and third. Option C is the right answer. Then we have the image shows a pollen grain from a grass plant. The image has a length of 60 millimeter. So the length here is 60 millimeter. That is the magnified value. The actual length of the pollen grain is 500 micrometers. We have actual size as well. 500 micrometers. Since you can observe that both of the units are different, we will convert them. So magnification comes out to be magnified value divided by original value. We have the magnified value as 60 into 10 to power minus 3 meter. We have the actual size 500 into 10 to power minus 6 meter. So this is 60 into 10 to power 3 divided by 500, and this comes out to be 600 by 5. That is 120. Magnification comes out to be 120 times. The diagram shows different type of cells. Which structure do all these cells have? So all of these cells have a cell membrane. Cell membrane is there, cell membrane is here, cell membrane is here, cell membrane is here. Though these two cells, basically this has a cell wall, this has a cell wall. Now coming to the options here, cell membrane present in all, cell wall present in two of them, chloroplast is present in the first cell, nucleus is present in first, third and fourth. And you can also identify that this second cell must be the RBC, since the mature RBC do not have you can say nucleus when it is mature. Option A is the right answer. Question number 17 says the drawing of the insect shows a magnification of 5 times. We have got the magnification here. So this comes out to be magnification equals to 5 times. Scale shown is in millimeter. So this is 0 over here. This is denoting to 125. This is 125 millimeter. So we have the magnified value as 125 millimeter. Now you have to calculate the actual size, and for that we have to apply the formula magnification equals to, and from now I will be writing just m equals to mv by av for that, right? It is magnified value by the actual value. Since we have to calculate the actual value, so we will rearrange the formula actual value equals to magnified value by magnification. This comes out to be 125 millimeter divided by 5, that comes out to be 25 millimeter. So option A is the right answer. All right. Moving to question number 18, that says the diagram shows a plant cell. Which type of cell is shown? So it is a columnar cell, and we have understood that in the specialization section of cells, we got the palisade mesophyll cell, that is a columnar cell. So we are taking this as an option that palisade mesophyll cell can be represented here. Now going for the other cell structures to confirm that it is a palisade mesophyll cell, we have the vacuole, cell wall, cytoplasm, chloroplast, nucleus, cell membrane. So all of these structures confirm that this is a plant palisade mesophyll cell. So we will write C as the right answer. Question number 19 says, in which cell structure does aerobic respiration occurs? And by that we mean the respiration that requires oxygen for the process to occur. All right, this happens in, we can guess the answer. So this happens in that particular cell structure which is able to produce energy, generate energy in the form of ATP, and that is for, we have mitochondria. And mitochondrion for single mitochondrion. All right, mitochondria for a plural term basically. This is A as the right answer. Which formula shows the correct way to calculate the actual size of an object? So I just told you that one question will be coming from direct this formula here. So we have the actual size as, or you can see the actual value as, magnified value divided by the magnification. You have to select the right option for that, and we have magnified value, that is the image size, and magnification in the denominator. So B here provides us with the right answer. I hope this is understood to you. All right. Then we have question number 21 that says, in a mesophyll cell where are chloroplasts found? So we have the cell structure here. All right. Let us recall the cell structure. Then we have got this cell membrane over here. We have this vacuole occupying up to 90% of the cell volume. Then we have the cell nucleus here. Then we will talk about where are the chloroplasts present. So these are here. All right. And what is this region where these chloroplasts are located? So label it along with me. We have this one is the cell wall. This one is the cell membrane. This region here is the nucleus. These structures are chloroplast. This region here represents vacuole. And you got this particular region, the thick liquid component of the cell, that is the cytoplasm. You can observe that the chloroplast is present in the cytoplasmic region of the cell. All right, between the cell wall and the cell membrane, incorrect. In the cytoplasm, B is the right answer. In the nucleus, in the vacuole. Just another question here. Question number 22 says, the cell shown in the diagram has been magnified 3000 times. We have got the magnification here. Magnification is 3000 times. Diagram is 21 millimeter, and you have already understood that by the diagram we mean that magnified value is present as 21 millimeter over here. So we will have to calculate the actual size. This is the actual size, and the formula for that comes out to be magnified value by magnification, that is 21 millimeter divided by 3000. Option B is the right answer. which features are possessed by all plant cells, and we have to compare cell wall with chloroplast as well. So cell wall is the characteristic feature of any plant cell. Whenever we talk about plant cell, cell wall will be there. Whenever we talk about chloroplast, so there is this condition that these cell structures will be present in those cells that are performing the process of photosynthesis, and it will not be present in all kind of cells. We have discussed this already. So considering root hair cell do not have chloroplast in them. So cell wall will be present in all plant cell, whereas chloroplast will be present in those cells that photosynthesize. We got the option as B. So B here is the right answer. Question number 24 says the diameter of an egg is 6.5 centimeter. So by the diameter of an egg, we mean that alright, so question continues and says the student draws the egg, diameter of egg in the drawing. So this is the magnified value and this is the actual size. So actual value here is 6.5 centimeter. Magnified value is 19.5 centimeter. We have to calculate magnification, that is magnified value by the actual value, that comes out to be 19.5 divided by 6.5 centimeter, that comes out to be 3 times. So option B is the right answer. Question number 25 says the diagram shows the structure of a palisade cell from a leaf. Which structure contains chlorophyll? So chlorophyll are present in chloroplast. We have to identify which structure represents chloroplast here. A here represents cell wall. B here represents nucleus. C here represents chloroplast, and we have got our answer. C is the right one, and D here represents vacuole. Alright, moving to next question. Question number 26 says a bacterial cell has a length of 40 millimeter when it is magnified by 20,000. So this is the magnified value here, that is 40 millimeter, and we have got the magnification as 20,000 times. Alright. Then you have to calculate the actual size. So for that we have magnified value divided by magnification, that equals to be 40 millimeter divided by 20,000, or you can also write it like 40 alright. This is 40 into 10 to power minus 4, considering the zeros are present in the denominator. This is 2. We got this as 20 into 10 to power minus 4, or you can say that this is 0.002 millimeter. B option is the right one. Alright. Then you have another question that says the diagram shows a flatworm. The flatworm has been magnified by 100 times. So you are already provided with the magnification, that is 100 times. You have got the magnified value as well, 50 millimeter. You have to calculate the actual size of the platform, and I hope you can solve these kind of questions very well. Alright, be precise with their calculations. So actual size, magnified value by magnification, this is 50 millimeter divided by 100. Now as you can all observe that the units are in micrometer. Now we have to convert this particular unit in micrometer. Alright, 50 millimeter divided by 100. So this comes out to be 50 into 10 to power minus 3. Alright, divided by 100. This is in meter. 50 into, you are going to multiply the denominator and the numerator both by 10 to power minus 3. Alright. So this is Alright, we have 5 into 10 to power minus 6 meter divided by 10 into 10 to power minus 3. You can solve it like 10 to power minus 6 meter divided by 10 to power minus 2, 5 into 10 to power minus 6 into 10 to power 2 here, that comes out to be 5 into 5 into 10 to power 2 micrometer. This is 500 micrometer. Let us cross check. So magnified value by magnification, 50 millimeter divided by 100. This is 50 into 10 to power minus 3 meter divided by 100. 50 into 10 to power minus 3 into 10 to power minus 3 divided by 100 into 10 to power minus 3. Alright. So we are left with 5 divided by 10 over here. In the numerator there is 10 into minus 6, 10 to power minus 6. On division we have 10 into 10 to power minus 3. So this comes out to be 5 into 10 to power minus 6 meter in the numerator, and in the denominator it comes out to be 10 to power minus 2. Alright. Yes, this is the correct answer over here. This is 2. Alright. Moving to question number 28, that says which features are possessed by all plant cells. So you have already solved this question and this is a repetitive question. So you can solve this on your own. You have question number 29 that says the length of a bacteria in a diagram is 50 millimeter. So since it has already mentioned that the length of the bacteria as present in the diagram, so this must be the magnified value. This is 50 millimeter. The magnification of the diagram is 5000 times. So we have magnification as 5000. We have to calculate the actual length, and for that we have the actual value equals to magnified value by magnification. Alright, so magnified value here is 50 millimeter divided by 5000 as the magnification. Since you can observe the options are having different units, you are going to convert the units again here. 50 into 10 to power minus 3 meter, 5000, 10 to power minus 3 on numerator, 10 to power minus 3 in denominator. So we did this kind of multiplication in the numerator and the denominator side so as to convert this 10 to power minus 3 into 10 to power minus 3 into 10 to power minus 6. That comes out to be you can relate it to like 1 micrometer equals to 10 to power minus 6 meter. So we will write the 10 to power minus 6 meter as 1 micrometer. So you're going to solve it like this is 10 to power minus 6 meter, 5000 into 10 to power minus 6, 50 micrometer divided by, all will get cancelled. This is 10 micrometer as an answer. Alright, I hope this is understood. This is 10 micrometer. So B option is the right answer here. Question number 30 says the diagram shows the structure of a palisade cell from a leaf. As you can all relate that this diagram is a columnar structure basically. Which structure contains chlorophyll? We have to identify that structure, which is chloroplast. Alright. Contained in chloroplast. So A here represents cell wall. We solved a similar question in previous slides as well. So B here represents nucleus. C is chloroplast. D is vacuole. We have got the answer as C, chloroplast. Question number 31 says, see how many questions have come from one topic only, that is the magnification, and those questions are directly formula based. A diagram of white blood cell has a diameter of 76 millimeters. So this is a magnified value of the cell. Value here is 76 millimeter. Then you have got the magnification. that is 8000 times. Then you need to calculate the actual diameter. So for that we have the formula as actual value equals to magnified value divided by magnification. That comes out to be 76 millimeter divided by 8000. Again you have to convert the units here since the answer is in micrometer. So we have 76 into 10 to power minus 3 meter into 10 to power minus 3. We have 8000 into 10 to power minus 3. So I have just did the same thing in one step here only. So this comes out to be micrometer. 76 micrometer divided by 8, that comes out to be 9.5 micrometer. I hope this is understood. Option C is the right answer. Question number 32 says, which row shows cell structure that are present in both root hair cell and palisade mesophyll cell? So talking about the cell wall, it will be present in root hair cell, it will be present in palisade mesophyll cell. Both of these cells are a type of plant cell, so this should be a tick. The chloroplast will be present in palisade mesophyll cell only and not in the root hair cell. This should be a cross. We clearly got the answer, but we will still continue with the other cell structures as well. So cytoplasm will be present in both of these cells. Vacuole will be present in both of these cells. Option C provides us with the right answer. Question number 33 says, so all of these questions are taken from past year papers. Let me remind you again. So basically we are solving the past papers and see how the questions have come in the past papers, and you are going to get a quick idea of the important topics that are very crucial as per examination point of view. Question number 33 says, which row shows the structures that are present in both root hair cell, and we have just solved this question as well. So it came in, all right. So here is question number 34 that says the diagram shows a cell at 1500 magnification. So we are already provided with the magnification here. 1500 times. Then we have the magnified value as, so this here is 4.5 centimeter. Magnified value as 4.5 centimeter. All right. What is the actual size of the cell? The magnified value as 4.5 centimeter, then the magnification is 1500. So this is 45 divided by 15 here into 1000. This is in centimeter. This comes out to be 3 into 10 to power minus 3 centimeter. 3 into 10 to power minus 3 centimeter. Then you are going to convert it 3 into 10 to power minus 3 into, so 1 centimeter has 10 millimeter, so into 10 millimeter here, this comes out to be 3 into 10 to power minus 2 millimeter, and by that you can solve that this gonna be 0.03 millimeter as an answer. All right, and we have got the option as 0.03 millimeter as option B. So B here is the right answer. Moving to next question, that is question number 35, that says which structures are found in the cells of all living organisms. So cell membrane will be there in all living organism cells. Chloroplast will be present in those cells that photosynthesize. Cytoplasm will be present in all kind of cells. We have 1 and 3. So option C here provides us with the right answer. Then we have question number 36 says a diagram of human red blood cell is shown. We have this diagrammatic representation. The length of line XY on the diagram is 40 millimeters. So XY here is the magnified value. So I'm going to write it here, magnified value 40 millimeter. The actual width of the cell is, so actual size is there 0.008 millimeter. What is the magnification of the diagram here, so that you can apply the formula magnified value divided by actual value, 40 millimeter divided by 0.008 millimeter, 40 into 10 to power minus 10 to power 3 here divided by 8. So this comes out to be 5000 times. Option B here is the right answer. Then we have question number 37 that says which statement about cell structure is correct? An animal cell has a cell wall. An animal cell never has a cell wall. A bacterium has a cell wall. They do have a cell wall. An animal cell has a chloroplast. So, animal cell never has chloroplast. This is also incorrect here. Then we have a bacterium has a chloroplast. A bacterium do not have chloroplast. Option B is the right answer here. Moving to question number 38 that says the photograph shows a limpet. The length of the line PQ on the image 65 millimeter is there. So, we have this magnified value as 65 millimeter. What is the actual diameter of the limpet? So for that we have to calculate what is the magnification, and magnification is provided in the diagram. We have magnification as 2.5 times. We can calculate the actual value, that is magnified value divided by magnification here. 65 millimeter 2.5 times magnification, that is 650 by 25. This comes out to be 26 millimeter. Alright, so this is 26 millimeter. I hope you got the answer. Option C is the right answer. Alright, then we have question number 39 that says which row shows structures that are present in both root hair cell and palisade mesophyll cell. So you can observe that how many times this question have come in the past years. Alright, so we'll skip this so you can solve this on your own. Question number 40 here says the photomicrograph shows a plant cell. The length of line XY is 90 millimeter. So we have got the magnified value here. 90 millimeter. Then you have got the actual length. So this is actual value 30 micrometer. Alright. What is the magnification? So for the magnification, you are going to apply the magnification formula here. That is magnified value divided by the actual value, and this is 90 millimeter divided by 30 micrometer. So you're going to convert these units. 90 into 10 to power minus 3 meter, 30 into 10 to power minus 6 meter. So this is 3 into 10 to power 3 meter, 3000 times. Alright, so you've got the answer as 3000 times as the right answer. Option B here is the right one. Question number 41 says which structures are found in cells of all living organisms. So we have already solved this kind of question. Cell membrane will be there, cytoplasm will be there. 1 and 3 is the right answer. In the diagram of a palisade cell, the distance between X and Y here is 20 millimeters. So here we have the magnified value as 20 millimeter. Actual width of this cell between X and Y, so we got the actual value as well, 40 micrometer. What is the magnification in the diagram here? So we are going to apply the formula as magnification equals to the magnified value divided by original value. Alright, we have got the magnified value as 20 millimeter. We have got the actual value as 40 micrometer, and we are going to solve it. 20 into 10 to power minus 3 meter, 40 into 10 to power minus 6 meter. This comes out to be 0.5 into 10 to power 3. This is 500 times. Option B here is the right answer. Moving to next question we have. Question number 43 says, which statement about cell structure is correct? We have already solved this kind of question. An animal cell do not have a cell wall. A bacterium cell has a cell wall. An animal cell do not have a chloroplast. A bacterial cell do not have a chloroplast. Option B is the right answer here. Question 44 says the diagram of an egg cell from a frog is 2.5 millimeters. So the egg cell of a frog is 2.5 millimeter. So this is, we have 2.5 millimeter. Then you have got the diameter of an egg cell from a human is, so human egg cell is 120 micrometer. Then you have to identify which value shows approximately how many times larger an egg cell from a frog is than an egg cell from a human. So you have to compare the frog egg cell with the human over here. You have the formula here as, you have to compare the frog cell egg cell with the human egg cell. So we will put the frog egg cell in numerator side, and on the denominator side we have human egg cell. Alright, so the frog egg cell is having 2.5 millimeter size, and the egg cell from human is 120 micrometer. Since the question was asking, you have to calculate by how many times larger an egg cell from a frog is. As you can also compare the units over here. So micrometer is a shorter unit and the millimeter is the longer unit or larger unit compared to the micrometer over here. So the frog egg cell is larger, whereas the human egg cell is smaller. So you have to compare by how many times the frog cell is larger when it compared with the human egg cell. This is 2.5 into 10 to power minus 3 millimeter. This is 120 into 10 to power minus 6 meter. We're going to solve it like, this is 25 divided by 1, 2 into 10 to power minus 3 meter divided by, so this is 10 to power 2 here into 10 to power minus 6 meter. This comes out to be 2.1 into 10 to power minus 3, 10 to power minus 4 on division. We get 2.1 into 10 to power minus 3 into 10 to power 4. This comes out to be 21 as an answer. So option B, that is 21 times, is the right answer to the question asked. Moving to next question that says the photomicrograph shows a cell from a type of aquatic plant. So aquatic plant also perform the process of photosynthesis. They do synthesize their own food from the raw materials, and they perform the process of photosynthesis by that. Which labeled part indicate that it is a plant cell. Alright, so plant cell have cell wall, plant cell have chloroplast, plant cell have vacuole. So we are going to find the labelings here first. So P here represents nucleus. Alright, then Q here represents cell membrane. R here represents cell wall. S here represents chloroplast, or you can say that the R here is extending toward inside, so we will label it like this is a cytoplasm. All right. This is pointing toward the inner side, and Q is the cell wall. Q must be the cell wall here. So Q and S. All right. We have got the answer as D. Q and S, that is due to the presence of chloroplast, due to the presence of cell wall, we can call this particular plant cell as a uh, a type of cell that has been taken from aquatic plant. All right, so Q and S. All right, moving to question number 46. That represents the diagram shows a magnified image of a cell. Magnification here is provided to you. So magnification is 360 times. The line LM here represents magnified value. So this gives 18 millimeter as an answer. We have to calculate the actual width. So actual value comes out to be magnified value divided by magnification. That is 18 millimeter divided by the 360 over here. So we got the answer as this is 1 by 20 millimeter, or you can also say 0.05. So this is the actual width of the cell. Option B is the right answer. So moving to question number 47. The diagram shows a red blood cell. We have the diagrammatic representation. So 49 millimeter here represents the magnified value. Actual diameter of the cell is 7 micrometer. So actual value here is 7 micrometer. You have to calculate the magnification here. For that we have the formula as magnification equals to magnified value by actual value. Magnified value is 49 millimeter. Actual value is 7 micrometer. So you're going to convert the units here. So this comes out to be 49 into 10 to power minus 3 meter divided by 7 into 10 to power minus 6 meter. This is 7 into 10 to power 3 meter. 7,000 times. Option C is the right answer. Question number 48 says root hair cells are found on plant roots. Which feature is present in a root hair cell but not in a sperm cell? So cell membrane will be present in both. Cell wall will be present in root hair cell but not present in sperm cell. Chloroplast is present in, so it will be not be present in a root hair cell since it is not performing the process of photosynthesis. Cytoplasm will be present in both. Option B here is the right answer. Question number 49 says a plant cell is viewed using a microscope with a magnification of 500 times. So we are provided with the magnification here. Image of the cell has a width of 20 millimeter. This is the magnified value, 20 millimeter. We have the magnification as 500 times. We have to calculate the actual value. For that, we have formula as magnified value divided by magnification, 20 millimeter divided by 500. All right. So this comes out to be 0.04 millimeter. Option A here provides us with the right answer. All right. Moving to question number 50. That says the cells listed have specialized structures that allow them to carry out their functions. So we have certain cells which cells have structures that can move. So ciliated cells, all right. So they have cilia that show the flicking movement basically. They move. Nerve cell do not move. Root cortex cell do not move. Sperm cell have flagella. So these are the two cells that have structures that can move. We have 1 and 4 as the right answer. Option B here is the correct one. 51 says a cell was viewed under a microscope. The actual length of the cell is, so we have got the actual value as 0.025 millimeter. Then you have got the magnification of the image 400 times. You have to calculate the actual length of the image here. So we are going to write magnified value as, this comes out to be actual value into magnification. Right, since we have got the formula as magnification equals to actual value, magnified value by actual value, so we can rearrange the formula accordingly. It gives the magnified value equals to actual value into magnification. We have 0.025 millimeter and magnification is 400 times. So this is millimeter. 10 millimeter is the right answer. Option A is the correct one. Moving to question number 52, that says, what is an example of a tissue? So this is a particular question coming from this chapter only, but this is not as important as the other topics, but we are going to solve one question at least for that, so we can recollect our information about that. And what is an example of tissue? So tissues are a group of cells that are similar in function and perform the similar function basically. So a chloroplast is a type of cell organelle and not a tissue here. The palisade mesophyll is there, a neuron is the cell, and the pancreas is an organ. So basically we have that as a tissue is a group of a group of similar cells performing similar function. All right, and for that, here we have chloroplast as the cell, neuron as the cell, pancreas as the organ. An organ is composed of many tissues. So basically, option B, the palisade mesophyll. So here we have many palisade mesophyll cells. So this will be an example of tissue. Option B here is the right answer. Question number 53 says the diagram shows a leaf which has been magnified at a magnification of 7 times. So we have the magnification as 7 times, the magnified value as 98 millimeter. This is 98 millimeter. What was the actual length? So actual value equals to magnified value by magnification. This is 98 millimeter divided by 7. Here we got 1, 2 point, that comes out to be 2.8 approximately. This is 1, then we have 2, 4. All right, so we have got the answer as there might be some error. This is 14 actually, so this comes out to be 14 millimeter. Option A, option B here is the right answer. Moving to question number 54, that says the diagram shows a cross section through a mesophyll cell of a leaf, which part is partially permeable, and we have already understood that this is a property of cell membrane. We have to identify the cell membrane from the given labelings here. A here represent cell wall, B here represent cell membrane, C here represent chloroplast, D here represent vacuole. So B option, that is representing the cell membrane that shows the nature of being partially permeable, is the right answer here. Alright, so by this we end the one shot for particular chapter here. And for further sessions like this, stay connected with us. Thank you so much. I hope this session helped you in many ways. Alright, so thank you. Meeting you in the next session.